Yeah. Hello. Am I still audible?
Yes, sir.
Yes.
Great.
You're audible.
Great. Yeah. So I see that recording has also started. So I mean, I'll just finish whatever is remaining from the last class, and then you can ask me questions in relation to that. Okay. Hold on a second. Yeah. So I need to close a couple of things here. Okay. This one I also don't need. Share screen. Yeah. Can you all see my screen?
Yes, sir.
Yes, sir.
Okay, great.
Yes.
Yes, sir.
Great. Okay, hold on. Just put it on now. Okay, so we were actually, where we cut off. Okay, before I start, please remind me when it's okay, and then if you don't remind, that's your problem, and I'm not looking at time or anything. All right. So we were talking about the hydrogen bond, and so hydrogen bond is one of the most important non-covalent interaction that you will find in proteins. Has been identified in early 1950s. Okay, and I'm sure that all of you are familiar with the hydrogen bonds. And for a hydrogen bond, what you have to have is you have to have an A, which is directly connected to a hydrogen, which is H, right? So AH, and there is sufficient electronegativity difference between the A and the H, and that AH bond, is in close proximity to another atom, which is B. Okay, so that B is also electronegative. Now, this is exactly what happens. For example, here you can see that. So imagine our A is a nitrogen and H is the hydrogen that is directly connected by a covalent bond. And that AH is in close proximity to B, which is an oxygen here. So, so here it turns out that you have an NH that is looking towards CO. And obviously you can imagine, as we also, briefly I showed you a table in the last class. So yeah, so it all depends upon the electronegativity. For example, we are talking about AH, right? So if AH is nitrogen and hydrogen, so the nitrogen electronegativity in the Pauling scale is like 3.0 and hydrogen is 2.1. So you have sufficient electronegativity difference between the AH, and that is again, looking towards an oxygen, which has an electronegativity of 3.4, right? So because of the difference between the electronegativity difference between the nitrogen and hydrogen, the bond is polarized, right? So the electrons are sort of withdrawn towards the nitrogen. So the hydrogen becomes more electropositive. And since the hydrogen becomes more electropositive, the sort of an interactive interaction between the more electropositive hydrogen and the oxygen, which is electronegative 3.4, meaning that it's sort of electron rich. There is the formation of I wouldn't say bond, but there is some sort of an attractive interaction. Okay, so this is a hydrogen bond. Now there are some interesting features about a hydrogen bond. For example, so if you have this AH looking towards, or let's say that this is an NH looking towards the CO. Okay, so the geometry of these three atoms, this nitrogen, hydrogen, and oxygen, is also very important in the formation of this bond. Okay, so if you have this nitrogen and hydrogen, which is pretty far from the oxygen, let's say the distance is 5 angstrom between the hydrogen and the oxygen, don't have a hydrogen bond. By analysis of crystal structures, people have realized that, and also you can do some spectroscopic experiments which will clearly show the existence of a hydrogen bond. And people have seen that the optimal distance is around 2.4 to 2.7 angstrom, right? This between the hydrogen and the oxygen. All right. It can be oxygen or any other atom. What I'm talking about, a hydrogen bond. The other also important thing that one must note is the geometry, right? So the angle between the, I mean, nitrogen, hydrogen, and the oxygen, or you can A, H, and the B. Okay, this angle is, or yeah, so you can imagine this and this, sort of this angle. Here it's close to 180 degrees. This angle is also very, very important. Okay. So typically people say that, if you have hydrogen bonds, they are sort of collinear, meaning that all three are in the same plane, but that's not true. Okay, because there are reasons which I will not tell you. Maybe you'll study in the chemistry classes in the later semesters. Okay, they're never collinear because there are something called as Pauli repulsion, because of which they can never be collinear. There is a specific orientation between the nitrogen, hydrogen, and the oxygen. Okay, there is a specific angle. All right. And it turns out that, so for example, this angle is also very important. So let's imagine that this NH comes out of the plane, okay, meaning that this NH is not in the plane of the paper, I mean, on the slide. Let's say that this NH is towards you. Okay, then the angle probably this nitrogen, hydrogen here, imagine the hydrogen is here, and the oxygen, it would be sort of an acute angle, right? Even if the angle is 90 degrees, you do not have the formation of a hydrogen bond. Okay, although you might have an appropriate distance. So this geometry and the distance, right? So meaning the angular and the distance criteria, that is sort of determining the presence of an hydrogen bond, indicates again that there is some sort of an orbital overlap associated with this hydrogen bond. It's difficult to only depict or portray hydrogen bonds in terms of dipole interactions or electrostatic interactions, because if it was just dipole or electrostatic interactions, you probably wouldn't need the angular and the distance criteria. I mean, distance criteria, yes, but the angular criteria probably won't be necessary. But since you have an angular criteria associated with a hydrogen bond, that means that there is some sort of a quantum mechanical nature orbital overlap involved in a hydrogen bond. Now, hydrogen bonds are very, very important in whatever biomolecules we will study, and you will study later in your biology classes or whatever. So, for example, so here, in this slide, can you see my slide now? The next one.
Yes, sir.
Okay, good. So here, you have a protein backbone, right? And the protein backbone, you will see later that there is a amide bond. So this carbon and this is an oxygen. This is a CO and this is an NH, right? And so typically, what happens? Imagine that when the protein is not folded. Now, what does it mean folded? We will see later. So imagine it's just sort of a straight chain of polypeptide backbone, meaning that there are multiple amides. Okay, in the case when there are multiple amides, and if this molecule is present in water, the water molecules can form hydrogen bonds, right? So the NH, because of the space available for the hydrogen, so it forms a single hydrogen bond with the oxygen of water molecule. And the oxygen, since you have two lone pairs of electrons available, it forms hydrogen bonds with two water molecules, right? Right. So in this way, the CO and the NH of a polypeptide backbone are involved in a hydrogen bond with the water molecules. But what happens, eventually when proteins fold, meaning that it's a linear chain, so let's say that there is a very long thread, and the thread sort of folds back on itself and takes some unique shape or whatever. What happens is these water molecules are essentially thrown out. Okay, and when the water molecules are thrown out, the polypeptide chain forms a hydrogen bond with its own polypeptide backbone, where you have the NH and the CO oriented towards each other in the specified, not specified, but the within the limits of the hydrogen bond angle and distance. And this way you can imagine that, see, initially if you are to calculate the number of hydrogen bonds, you will see that this CO and the NH were involved in more number of hydrogen bonds with water molecules than after they take a nice three-dimensional shape. So there is, out of three hydrogen bonds, two are lost, and it just forms a single hydrogen bond, right?
Sir,
So
I just have a doubt, sir.
yes.
Are these two different proteins forming hydrogen bonds with different
No,
water molecules, or
we're
is
talking
it the
about the
same
same.
protein?
No, we are
Oh,
talking about the same polypeptide chain.
oh, sure, sir. Thank you.
Yeah. So I mean, so imagine it's just a 20 residue polypeptide. Okay.
And I also have a doubt in the previous slide that you said that they shouldn't they are it is not necessary that the thing should be collinear, and then you said that they should it is not necessary that they should be in the plane. So
Hmm.
do you mean collinear or coplanar?
Yeah, it's not. Yes.
No, no. Did you mean coplanar or collinear?
I believe that if you have 3 points and they are collinear, they would be in a plane anyway, or not,
Yes, yes.
isn't it? Yeah. Okay. So yeah, so I mean, it's not necessarily meaning that now it's known. Earlier, it was not known that a hydrogen bond, they're these three atoms, that A, H, and B, they're never collinear. They're never, uh, because there are repulsion factors associated with the hydrogen bond that does not allow them to be collinear. Okay, so rather there is a specified geometry, I mean the angular and the distance criteria at which you get the strongest hydrogen bond possible. Okay, yeah. So here, so this is the same polypeptide chain. Imagine this is a 20 residue polypeptide, and we are talking about the folding of the same polypeptide. I mean this specific 20 residue polypeptide. So you can imagine that each NH is forming one hydrogen bond with one hydrogen water molecule, and COs are forming hydrogen bond with two water molecules. But when the polypeptide folds back, and if it forms hydrogen bonds with the backbone COs and the NHs, then so out of several hydrogen bonds with water molecules are essentially lost. And so if you are to calculate energetic difference, you will see that so there are a lot more energy associated with this unfolded form in terms of hydrogen bonds, right? Than in this case. Okay. I mean, if you're talking about a specific NH, but you can see that, for example, this CO is involved in hydrogen bond, but this CO is probably still involved in some sort of a hydrogen bond with water molecules, right? So there is a nice balance, and some are lost and some are not lost and all. But yeah, so you have hydrogen bonds. The other thing also one must understand, and I want to tell you, I don't know whether you've studied, but it's not really necessary that you have to have that A, H, and B kind of atoms for the formation of hydrogen bonds, right? You can also have A, H, and A, right? So here, you can see that this is you can imagine this is well, it could be aromatic ring here, okay, and the nitrogen is in a, well, it could be a pyridine ring, for example. In a pyridine ring, the nitrogen is involved in the aromatic ring, and here it can be some other NH, right? And this NH forms a hydrogen bond with N, right? So this N, there is a H, and A. Well, yeah, and why does it form so? Because again, since this nitrogen is connected to two carbons, right? And if you look into the electronegativity difference of this bond and that bond, there is still a difference, right? It's probably less, but you definitely have an electronegativity difference. For example, we are talking about nitrogen, which is 3.0, and we are talking about a carbon, which is 2.6, right? So you still have a difference, zero point 0.4, but there is a difference. So that means that there is certainly some sort of a dipolar character of this nitrogen, and that's why this is more delta negative, right? And this is obviously, there is no question about this dipole, and they form a hydrogen bond, although the hydrogen bond formed here probably would be a lot weaker than if you're comparing it with a NH and a CO kind of a hydrogen bond. Okay. And the other thing, so this is a weaker hydrogen bond, but nevertheless, it's a hydrogen bond. And there are also certain hydrogen bonds which are really, really weak. For example, so if you have, instead of a NH here, so imagine you have a carbon and a hydrogen, okay, just a CH, right? And these CHs, right, can form very weak hydrogen bonds with electron rich centers. For example, you can have an aromatic ring like tryptophan. You will see what is tryptophan later. Tryptophan is an amino acid where there is an indole ring. Well, indole is a, I don't have the structure here I could draw, but yeah, we'll draw it later. Well, let's say that you have a system which is extremely electron rich, so electron dense, right? So if you have a very electron dense center and you have a CH, okay, well, sometimes it's necessary that this carbon is also connected to, it could be connected to an electronegative. For example, this carbon could be connected to a sulfur or maybe a nitrogen. Okay, so so here I can show you. For example, yeah, here. So in this one, this is an amino acid where this carbon is called as a alpha carbon. Okay, we will see later what are alpha carbons. So this alpha carbon, since it is directly connected to a nitrogen, this is more electropositive than a CC bond. Okay, this bond is a little polarized than a regular CC bond, and obviously because of this, since the electron cloud is sort of withdrawn over in this direction, this hydrogen is more electropositive than ethane, let's say. Okay, and so it gives more electropositive character to this hydrogen. And since this hydrogen becomes a lot more electropositive, if in the vicinity there are some electronegative groups, for example, as I said, that aromatic rings or whatever, then those aromatic rings can engage in some sort of a very weak hydrogen bond kind of interactions. Okay. So, but again, the energy it all depends. It depends upon the solvation state of this molecule and other features. Okay, the other thing that I also want to tell you that so now this is a reversible process, right? So you can see that in this way, the protein, when the protein folds from going from the left to right, the polypeptide forms hydrogen bonds with itself, and the atoms and the groups come in proximity to each other. But when it goes in the reverse direction, right? So in the reverse direction, when the folded to the unfolded state, what happens is it actually, well, it's shown that, for example, if we have intervening water molecules, then what happens is, so imagine if you give access to water molecules in this space here and this space here, the water molecules, the oxygen of the water molecules will compete with this CO to form hydrogen bond with this, right? So in this case, what happens is the net, well, let's say if it's not a covalent bond, right? So it doesn't have to be constant. It's sort of flickering, right? And since it's flickering or fluctuating, there is always a finite possibility that NH forms a stronger hydrogen bond with this OH here, this water molecule here, which if you give access to a water molecule, and if this happens, the hydrogen bond which is formed between the residues of the polypeptide chain gets weaker. And if it gets weaker, the protein will essentially lose its three-dimensional structure. Now you will see later that when a polypeptide folds and forms a very nice compact structure.
Repeat once this part. Please repeat once this part.
Yeah, sure. So what I'm saying is, imagine that there is a protein. I'll show you a cartoon and then you can probably appreciate. Yeah. So imagine this is the protein, right? This yellow bean-shaped structure. This is the protein. Okay. This is not a two-dimensional thing. Imagine there is a three-dimensional feature to it. Now what happens is the surface of the protein is exposed to water. Okay. But the interior, right? The interior of the protein, it doesn't necessarily, not necessarily, in most cases, these proteins, which are called as globular proteins, they don't have access to water molecule. Now, the reason is, if you give access to water molecules in the core of the protein, what will happen is this. So imagine now So in that yellow bean three-dimensional structure, you have this thing which is present in the core, right? When it is present in the core of the protein, that means there are no water molecules present, okay? Only this hydrogen bond is present. But imagine that you can put a narrow bore into that protein and put some water molecules inside. So what will happen is that when the water molecules come in, they will compete to form a hydrogen bond with this NH, right? Like what is happening here, right? So that means that the NH is not just shared with the CO of the polypeptide chain, but the NH will also be shared with the water molecules that have gained access to this. Now, if this happens, what happens? This hydrogen bond will get weaker, and if this hydrogen bond gets weaker, the structure of the polypeptide will loosen up, or let's say the core of the polypeptide will loosen up, and it can probably go to the folded state, to the unfolded state. Is it clear now?
Yes, sir. Thank you, sir.
Yeah. So so this is okay. Now in terms of energy, how are we doing with the energy of a hydrogen bond? If you are to compare the energy of a hydrogen bond with a ionic interaction, like some sort of salt bridge, like glutamate and aspartate that we discussed last time, and a very weak non-covalent kind of interaction where two non-polar, non-charged atoms are interacting. Okay, so here in this graph you have three curves. So obviously in your y-axis you have the energy, it is the energy axis, and this is the distance axis. And you can see that, so we were discussing about the van der Waals radii, and this is the van der Waals curve. The black one is the van der Waals energy, okay? And where the energy is very, very low, it's around 1 kilojoule per mole, maybe around 2.24, 2.25 kcal per mole or something, okay? Now, if we are talking about the electrostatic interaction, the electrostatic interactions are a lot stronger. They're of the range of like 20 kilojoules per mole. But if you're talking about hydrogen bonds, well, it also depends upon the groups involved. It depends upon, as I said, that the geometry of the hydrogen bond. It also depends upon the access of water molecules. Okay. So with this, what you can essentially see that the energy of a hydrogen bond is around so minus 10 kilojoules per mole. Okay, so yeah, so you can see that you probably from this graph you can appreciate that the strongest ones are definitely the electrostatic interactions, and then followed by the hydrogen bond kind of interactions. Okay, it's typically said that this hydrogen bond interactions, it can be put to 2.5 to maybe 5 kcal per mole, sort of. Okay, you can convert that to kilojoules per mole also. And the van der Waals is around 0.24 kilocalories, 0.25 kilocalories per mole. All right. Yeah, so this is all about the forces. I mean, there are many other forces in proteins. Now, when we study the biomolecules in details, I will try to touch upon those specific points where you can appreciate, why the forces are important, what sort of non-covalent interactions are there, why hydrogen bonds are important, right? Yeah, so also, you're probably familiar with the standard nomenclature that, the atom, the AH, which actually has the hydrogen, which is directly covalently bound to the atom, is called the hydrogen bond donor. And the one which is engaging in a hydrogen bond, which is non-covalently bound, is called the hydrogen bond acceptor. So here, this
Sir,
is the
I had a doubt in this portion.
Yes. Yeah, sure.
Yes, sir. Here you said that the total number of hydrogen bonds is decreasing. So does this translate to the fact that if proteins fold, then somehow their solvation sort of decreases or something like that?
Oh yes, that's a very good, very good interpretation. Absolutely, yes, absolutely. You're very right. When proteins fold, their overall solvation definitely decreases, and you can actually, well, people have, from year, I mean, since maybe 1970s, 80s, people have, many people have, tried to understand the protein folding process, and they have realized that when proteins fold, obviously, there is heavy desolvation of the amino acids, And every amino acids, as we will see the architecture of the amino acids later, you will see that every amino acids has certain contribution to the desolvation. Okay, so meaning that amino acids which are composed of completely hydrophobic amino acids, hydrophobic residues in the side chain, they contribute lot more to the buried surface area or the desolvation, and the amino acids which are lot more polar, their contribution to the buried surface area or the desolvation is lot less.
Excuse me, sir.
Yes, sir. Yes, sir.
Okay, yeah. Now you can ask me any questions related to this chapter, because once the questioning is done, and then I can go to the next chapter,
Sir,
which will be lipids.
sir, the water molecules,
Yeah.
sir, the water molecules around the protein
Hmm.
that gives the solvation energy,
Yes.
and then the hydrogen bonds are mixed up, and those proteins form intermolecular hydrogen bonds. And that hydrogen bond can be broken when water interacts again, and then the energy is further released.
Hmm.
Means there is only mix of energy on right hand and left hand side. Means
Hmm.
there is no overall release and no overall gain of energy.
Hmm. Hmm.
Is it so?
That's very nice, very good. Yes. So I mean, yes, you're right. See, that's why this has been a subject that for protein folding, not for protein folding, for many biomolecules folding, that in terms of the, if you think of the net contribution of the hydrogen bonds, the net contribution of the hydrogen bond does not is not enough to justify that why a protein should fold, right? Meaning that it goes from the left side to the right side, because what happens, you will see that in the initial case, when the polypeptide is unfolded, the polypeptide is forming hydrogen bond with water molecules, and when the polypeptide is folded, you will see that the polypeptide forms along with some of the groups which are forming hydrogen bonds with water, like in this carbonyl oxygen here. Some of the NHs are involved in the formation of a hydrogen bonds with its partner CO, meaning that from the same polypeptide chain. So in essence, if you are to calculate net energy based on the contribution or the involvement of the hydrogen bond, irrespective of whether it is with water or its own residues, you will see that there is not a huge difference. Okay, so so that essentially concludes that, or people concluded that hydrogen bonds do not contribute to the protein folding. There are other forces which actually contribute to the protein folding. It's a
Sir, this phenomena can be considered like the benzene part, which the benzene, we don't know whether that double bonds are there. So is it like that? In this case, we don't know whether it is folded or not folded. So we're just assuming it is folded also and unfolded at the same time.
No,
Is it like that?
No, I didn't understand your question. No.
Sir, like the benzene part, sir, we consider the overall structure to be like electron rings, electrons distributed. So is it like this, that the protein are free and also bonded at the same time, like here?
No, so you mean to say that, is that a speculation that we are thinking that protein is folded and unfolded at the same time? Is that the question?
Yes, sir.
No,
like
no,
we don't know.
no.
We don't know whether it is folded or unfolded.
It is.
We're
We
just
know it is
considering
folded. No, we know
folded
it
and
is folded.
unfolded at the same time.
No, no, we know. We know. If you are given a protein in a tube, you will definitely know there are several spectroscopic and many other techniques using which you can accurately determine, not only determine, you can say that how much of the protein is folded. You can also determine its population, how much of that is folded.
Thank you, sir.
And you, not only that, you can also determine the precise structure of those. Okay.
Sir,
Thank you.
When an
sir,
egg sir, when an egg coagulates, then when it is heated, the hydrogen bond should actually break, and when the hydrogen bonds break, and the protein should actually get unfolded. So its solvation would then increase. But then on heating, the egg coagulates. So how is this contradiction?
Are you suggesting that how am I stating it just based on the solvation? Because you are saying, or as I said always, that if a polypeptide chain is sort of flexible, then it's more solvated. But when a polypeptide chain is folded, then it's desolvated. So, are you asking that then how is it justified that the number of hydrogen bonds cannot are not useful in determining in terms of energy the folding of the protein? Is that the question, or did I get it wrong?
No, I meant to say on heating, the hydrogen bonds should break.
Correct.
So
Yes.
the protein will get unfolded.
Correct.
And when the protein gets unfolded, its solvation will increase.
Correct. Yes.
So when we boil an egg, why does the protein solidify instead of getting more liquid?
Okay. That is a completely different phenomenon. Okay, that is called as aggregate. Okay, that is not a folding and unfolding. Okay, that is something else. That I will, when we talk about, I'm not going to specifically talk about the, boiling or the making of an omelette, but that is essentially, what happens is, for example, it's a very similar thing that you can argue that many people, well, females typically they have curly hairs and they go to saloon to straighten their hairs up, right? And that is, you can do a temporary kind of straightening, you can do a permanent kind of straightening, and the temporary kind of straightening comes from heating those up, because what happens heating those up, I think the collagen or something, whatever keratin, what is present there, they lose their structure, and since they lose their structure, the hair straighten up. But eventually, as you have more and more new proteins accumulation, those are essentially removed and the hairs curl up. And there are certain ways of permanent denaturation also. So what you're doing here in a case of an egg is you're completely denaturing the protein. And by denaturing, you are probably taking those polypeptide chains into states where it's not just the solvation anymore. It's more about, they are stuck in a state where if you solve it, first of all, you probably cannot solve it, okay, because what has happened, the polypeptide has
There's some
completely
background
lost its
noise.
structure.
I can't hear you.
Can someone else turn off their mic? Hello?
Yes, sir. Now you are audible.
All right. See, what happens is, so that's not the question of a solvation now in eggs, right? That is a denaturation. So what happens is, so in that case, you have the polypeptide chain, and the single polypeptide chain, it's sort of frozen in a state where you cannot solvate it just by adding water, okay? Because what essentially has happened is, many polypeptide chains have come together and have formed a very rigid sort of structure which completely excludes water and allows the refolding of the protein.
So in this case, are we considering just one protein?
Yes, of course. We are considering one protein. Yes, we are not. We are completely ignoring the concept of aggregation at this stage. All right.
Sir,
Yes.
some moments ago, the question that was asked, I didn't understand it exactly. Was the question something like the energy difference between the reactants and the product is very less? So how is it favorable, the protein folding process?
Yeah. That's
So, sir, is like in biological systems, since it's not that favorable on the energy basis, chaperones are involved in this.
No, I know. Well, okay. So you have studied about chaperones?
A bit. I read them in class 12.
So that would be energy rich process, right? For that, you wouldn't require ATP.
Oh, yes, yes.
Okay. So here we are not talking about, some energy rich processes. When we talk about protein, we'll talk about a beautiful experiment by someone called as Christian Anfinsen. You will see that, he showed that if you just have a polypeptide chain and you do not give energy, you just, keep the protein in some sort of a denaturant or non-denaturant, the proteins can completely, either fold depending upon the condition, fold back to its native state, or unfold if you have denaturants there. Okay.
Okay, sir. Thank you. Okay.
I mean, just talking about the intrinsic properties, nothing really aided like chaperones and all. Okay.
Hello, sir.
Yes, yes, yes.
So you said that the proteins actually, the water actually competes with the
Yes.
other atoms of the protein. So to like actually competes with the process of the folding of the proteins. So what if the amino acid have very hydrophobic or sterically hindered groups like phenylalanine, like very big groups? Then what will be the like their interaction with the water?
Yeah. So essentially, you will see that hydrophobic effect is the one that actually promotes the folding of the protein. So okay, so if you have those non-polar residues, they actually come together and sort of exclude water and promote the folding of the protein. Okay, but if you are talking about just a polypeptide chain and thinking that I have two phenylalanines here, right? Man, two phenylalanines are enough to actually fold the protein. That is not going to happen. Okay, so it's not going to happen that if you it's not like a cascade kind of reaction where you just remove or desolvate one specific amide bond if it forms some sort of an intramolecular hydrogen bond, and that sort of promotes or catalyzes the formation of the other intramolecular hydrogen bonds throughout the protein if it's a very, very polar molecule. Okay, because you will see that later. I'm not going to talk about here. There are certain proteins which are called as intrinsically disordered proteins, and you will study when you study about intrinsically disordered proteins. I don't know whether you study or not, but when you study those, you will see that these polypeptides are composed of very flexible and polar residues. Okay, so since they are composed of very flexible and polar residues, it's very, very difficult to have some sort of an hydrophobic effect and desolvate the polypeptide bond, okay, to form a globular structure.
Okay, sir. Thank you.
Sir, in the last lecture, you mentioned that to prevent the binding of the virus with the receptor protein, we need a neutral molecule, neutral inhibitor. But why is it necessary to for the inhibitor to be neutral? Because the protein may also have.
Did I really say neutral inhibitor? I can't remember I said neutral inhibitor. I'm not sure. What is your question?
So actually, when you mention neutral, you meant that it does not, it's not like a virus or it does not attack the body. It's neutral as in it has no effect on the receptor.
Oh, okay.
Yeah, so not in terms of charge, but in terms of the functionality. That's probably it. Yeah, good. Thank you for helping. Okay, anything else? I mean, even from the last class. If not, then I will slowly go into lipids.
So in the case of the hair, which you told that the reforming and the deforming and the reforming of the curliness of the hair,
yeah,
isn't it related to the disulfide bonds, the breaking of the disulfide bonds in the keratin protein?
Correct. But what does the disulfide bond do? I mean, disulfide bond leads to the folding of the protein. You will see later. Disulfide bond is so. For example, okay, so I mean, those are so the that I if I'm not wrong, I think that is a treatment where you really lead to either a permanent or a semi-permanent kind of or let's say the long-term loss of the curliness, if I'm not wrong. But when you heat it up, by heating it up, you can also get it straightened. I don't know how efficient it is, I've not done research on this. So essentially what happens is just denaturation. The loss of the disulfide bond is also leading to denaturation, but you can imagine, since disulfide bond is a covalent bond, the protein polypeptide chains probably are still hold close to each other enough that they come close and start folding on their own. But if you do not break the disulfide bond, but break the other bonds like hydrogen bonds by heating it up, you can still lose the structure.
So that means there are a lot of factors into that.
Yeah, sure. I don't know. I do not know the specific of the how exactly it is done, what chemicals are used. But if I am not wrong, someone told me a long time back that you do use certain reagents which leads to the breakage of the disulfide bonds for the for the long-term loss of that curliness or whatever.
Oh, so and another thing, like I wanted to know, like if we can see some reference text to the like chemical evolution, like the slides which you are showing in the class last time. So can we look more into those reference texts? Like if you can say something on the reference text.
You mean
like if
that
we want to go deeper into those?
I can, yeah, I don't have those. I have to find those. I can send it to you.
So if you can upload that in the Teams chat, it would be
You
helpful.
mean the specific
Yeah,
paper
the chat in the Teams
discussed?
chat.
Yes, sir, that would be helpful.
Those will be research papers, okay? That should be okay, because there
sir.
won't be textbooks. All right, but okay,
Yeah,
I'll
all
add
right, sir.
I'll add some material, some fun reading material where you can read and satisfy your enthusiasm. Okay. All right, so I'll go to the lipids part now. Hello.
When the proteins are denatured in solution,
Yeah.
will the solubility increase, or do they bundle up and it will decrease overall?
One second, please.
When this proteins are denatured in solution,
Yeah.
due to increase in hydrogen bonds, do the proteins increase, like their solubility increases, or do they bundle up and decrease the overall solubility?
No, I mean, yes. So that's yeah, that's a good question. I think typically what happens is you can imagine that the solubility will increase. Yes, definitely the solubility will increase because just from a very coarse perspective. So you have a 20 residue polypeptide, right? And when it's not structured, let's say there are 19 amide bonds or 18 amide bonds. So those 18 amide bonds are involved in hydrogen bond with water molecules. Okay, so intermolecular hydrogen bonds. But when this 18 or 19 residue, 20 residue polypeptide folds, out of those 18 polypeptide which are forming intermolecular hydrogen bond with the water molecule, maybe 8 or 9 or I don't know, some XYZ numbers are involved in the formation of intramolecular hydrogen bond within itself. Times what happens is, well, many times what happens is, well, again, it's a very general case. You can catch me on a very specific protein for which the solubility could be high in a folded state. In general, I'm considering a polypeptide, which has reasonable distribution of hydrophobic residues. So with that, what will happen is some of the water molecules will be essentially thrown out, and it will be forming intramolecular hydrogen bonds, right? So obviously, I believe that the solubility will decrease because those hydrophobic patches, I mean, there will be hydrophobic patches around the protein also. Okay, so meaning that it's there are certain regions which will be more hydrophobic, and well, it's a very complex thing to answer, and many times what happens if you have hydrophobic patches, those hydrophobic patches will prefer to come close to another hydrophobic patches. And then what happens is there is only to a specific limit you can concentrate the solution. Beyond that, if you do, these proteins, because of those hydrophobic patches coming close to each other, they will start aggregating. But maybe in a linear form, this may not happen. It's a difficult question to answer. I mean, but do you just did you understand what I tried to explain?
Yes, sir.
Okay, hold on. So now let's. I just have 2 minutes, but I can start this. Yeah.
So the ribosomes are also composed of RNA and protein, but they are not associated with any membrane. But they may not be associated with any membrane. So how come they are stable if on folding the solvation decreases?
We'll talk about these things later, okay? Because otherwise, these questions are so exciting and intriguing that we can have 5 hours session and still we won't be happy with the answers. Okay, I'm sorry. So we have to continue, and then we can have. You can ask the TAs. They are there. Oh, by the way, last week there was no TA session because there was a confusion. But you can ask this question to the TAs. The TAs, one of the TA, I mean, both the TAs work with proteins, and they would know. One of them work with membrane proteins. The other one, I think, I don't know what Sarah works on precisely, but I'm sure they can answer your questions extremely well, much better than me probably. Okay, so the next thing that we are going to start are lipids. Okay, because lipids are one of the simplest biomolecules that you will see. Can you see my slide?
Yes, sir.
Okay, great.
Yes, sir.
So lipids are one of the simplest molecules. And it's very interesting. These lipids are essentially those molecules which are not water soluble in a single molecule level, or let's say at a single molecule kind of level, but they are soluble in non-polar organic solvents. Okay, these are essentially lipids. They're very interesting features of lipids. Now, what you see that, okay, so these are hold on, let me get the laser pointer. Yes, so these are single-tailed lipids. Okay, I mean, we'll go to the detailed chemistry of those in a few slides. But what you can see that you have a sphere. This sphere essentially represents a polar head group. Okay, and this this wavy thing, this long slender thing that you see here, this represents a non-polar tail. This is typically composed of hydrocarbons. All right, and these are single-tailed lipids. Okay, meaning that you have just one head group and then you have a single tail. Now, the other thing that you have is, so you have the same head group. Well, the head group could be a little larger. Okay, and this head group, one single head group, is associated with 2 tails. All right, and it's very interesting to note that if you have this kind of lipids where you have just a polar head group with a single tail, these guys form structures which are called as micelles, right? And of course, you need a certain concentration of these guys in water to form these micelles. However, it turns out that if you have lipids, which are composed of 2 tails, right, 2 hydrocarbon tails, these guys do not typically form micelles. Instead, what they form is something called as bilayers. Okay, meaning that you can have the formation of planar bilayer. Okay, so you have these are essentially like I mean, I hope you understand. It's just like a piece of a cake. Okay, so these are lipids, and they also form very nice spherical vesicles. Okay, so this is a cross section area of a vesicle. So you can imagine that so in this case what happens is so outside there is water, and inside, of course, these all the hydrocarbon tails are present where the water is completely excluded. Okay, but in here what happens is you have so this one is let's say, yeah, so a ball, a tennis ball essentially. Okay, so in a tennis ball, imagine this is the outer surface. Okay, and this is the inner surface, which will be very thin in a tennis ball, and inside the tennis ball you have a cavity. Okay, and this is certainly the cross section area. Okay, the cross section, the vertical cross section of a tennis ball. And so inside of this sphere you have water. Okay, so this vesicle has engulfed in water. Okay, and this is something you can imagine that you have in mammalian cells or the protocells that we were talking about in the evolution thing, right? So this will be more like those. Now the question is, why do what essentially determines that, you know, if you have two tails, these guys form lipid bilayers, and if you have a single tail, it forms the micelles. Does anyone know? Does anyone have any clue?
Sir, can you repeat the question?
Yeah. So my question is, if you have given the same head group, if this guy has just a single tail and the same head group but two tails, this guy forms such architectures which are lipid bilayers. But if you have a single tail, these lipids form micelles.
Sir, I don't know the exact answer, but like one idea that I have is like usually what we have in lipids is that like the head group is something like glycerol or something like that. So if we put one chain, then we have two free OH groups that can interact with the water all around. And if we put two chains, then we have only one free OH group. So like the relative amount of solvation will decrease. So micelles will be more stable for single chain lipids and less stable for double chain lipids. So double chain lipids will try to form a more compact bilayer or something like that.
I didn't understand, because essentially, irrespective of whether you have a single OH or two OH free, the head groups are solvent exposed anyway.
Yes.
So how does it determine the architecture?
No, that was so the relative amount of solvation for single chain lipids is slightly greater.
So okay, so yeah, agree. Yeah, agree. So how is it related to the architecture? Because in the cases you see that water molecules are present.
Sir, I think he means to say that if we have one single chain, then the surface area is say x, and if we have exposed to water, and if we have two tails, then the area is less than 2x. So relatively less area is exposed to the hydrogen bonds to act.
Okay.
And because even if it is hydrophobic, it may consist of glycerol. So the relative solvation is less.
So
it is less than 2x.
So you mean associated with the packing or what?
Yes. That's what I was trying to say. Like the packing would change depending upon the relative amount of solvation that happens.
Okay, fine. Maybe that has a role. How about the tails?
Sir,
Like the tails might interlace, or are they like?
Yes.
Can the tails interlace?
What interlaced?
Yeah.
I don't understand. What is interlaced?
Like can the like?
Sir, can we
I don't
say
know.
Sir, I'm not sure if this is
spherical structure. Sir, can we say that in the spherical structure, the density of tails would have been very high if two-tailed lipids were to form micelles. So, in order to increase the volume, a sort of a bigger shell-like structure is created, which is the lipid bilayer.
Yeah, it's related to that.
Increase the volume and to accommodate.
It's related to that.
But wouldn't that be
So I think it gets more surface area to form double-layered micelles.
Correct.
So since it has two chains in the lipid, so it gets more number of it gets more surface area to form. Maybe like that. I'm not sure.
No,
but
surface area would
the
not be
double
very
chain
also, sir, because of
wouldn't
the
double chain lipid?
also, sir, because of the double tail, it gets bulkier, so it doesn't exactly fit in the micelles. So I think that is what Gurkirat also said.
Yeah. So this is related. Yeah, this is related. So what happens is, so if you are to compare the cross section area It all depends upon the cross-section area of the sphere and the cross-section area of the tail. Okay, so you could probably imagine these single-tailed ones as like ice cream cones. Okay, so so you have ice cream cones with a single or double scoop of ice cream. Okay, yummy. Well, it's risky to eat those at this time, but nevertheless, if you have several of these ice cream cones, so they will sort of resemble this, right? So the top part is larger than and a very narrow sort of tail. Okay, so if you are trying to bring all of these guys close to each other, because you can imagine that the head group is only going to interact with the head group and the tail is going to interact with the tail, right? So if you try to bring these guys closer and optimize the structure, you will, irrespective of what you do, you'll essentially lead to it. If you try to compact, right, the compactness will be such that all the hydrophobic tails will be hidden from the water, right? These micelles are, it's a two-dimensional structure, but if in a 3D, if you can rotate it, this is what you're going to see from all the areas, right? So it's like a cone, right? You try to get all the cones together to form a sphere. But the problem is when you have these guys, right? You try to bring all of them closer. It's more, instead of a cone, what has happened, the bottom part is also, if not equally thicker, but considerably thicker, right? So they resemble more like a cylinder. And if you have, well, with some certain amount of curvature or certain amount of disorder, whatever, okay? And if you try to bring them closer, they're not going to form a cylinder. They're not going to form a micelle, right? Instead, they will form this kind of structures. Do you understand that?
Yes.
Yes, sir.
This is exactly that.
Sir, in double-tailed, we have bottom considerably thick, and in the single-tailed, we have narrow tail. So
yeah,
that is responsible for.
That's right. That's right. So it's like a, so imagine you have a steel cone kind of thing, and those steel cones, you can try to pack them very nicely into a sphere, but if you have steel cylinders, very tiny steel cylinders, you cannot pack them into a sphere, right? You will either pack them into like this, like very like a lipid bilayer, which is linear, planar, or some sort of a sphere if there is a certain if they're a little narrow, because I mean, they are not like steel or hard bodies, they are rigid bodies, they are flexible, right? Okay, so yes. Now, yeah, so what is the typical architecture of these lipids that are typically found in biological membranes, right? And the lipids that are found in biological membranes, they are actually called as glycerophospholipids. Okay, now what does glycerophospholipid mean? So meaning that you have, the core is basically glycerol. Okay, so this is CH2OH and this is CHOH if there was no ester group here, right? And this would be CH2OH. Now what happens is, one of those primary alcohol is esterified by the formation of a phosphate using a, so you have a, you have a phosphate group attached here. Okay, it's a phosphate ester, and the other two, the other primary and the secondary alcohols are esterified by the fatty acids. Okay, and so this is the typical architecture. And what happens is you also have a pendant group, this R group here, with one of the alcohols, one of the OH of the phosphate group. Okay, and this R group, in many cases, it varies, right? And for example, this R group can have a choline residue. This is called as a choline, right? So meaning that this part is connected to the oxygen. Okay. So then this molecule, this entire molecule would be called as a phosphatidylcholine, right? You can also have this ethanolamine, which is connected, which is present at this R. Then this molecule would be called as a phosphatidyl-ethanolamine. Okay, and if you look into the charge on the lipid, since the phosphate group already has one negative charge, if you have a positively charged R group, what happens is you get a neutral molecule. Okay, So this phosphatidylcholine and phosphatidylethanolamine both are neutral. But then if you have a serine, now serine is an amino acid. If you have a serine, this positive and a negative charge essentially neutralizes each other. You have one negative charge. So this is a net negatively charged phospholipid. You can also have glycerol. Okay, you can have another glycerol here. All right, then this is called as phosphatidylglycerol. You can also have complex molecules. For example, these are well, you can see that this is a cyclohexane ring, right? You might think it's a sugar molecule, but it's not a sugar molecule. When we go to the carbohydrates, you'll see that this is not a sugar molecule. Many of you, this is a cyclohexane. This is a boat form of a cyclohexane, right? And this has several OH groups. This molecule is called as an inositol. Now, if you have this inositol present here, this molecule is called as a phosphatidylinositol. Now, why is it important? So depending upon the R group, what happens that you have the charged glycerophospholipids, and the charged glycerophospholipids are sort of determine what sort of interactions these lipid bilayer will have with different proteins that are coming from the outer environment to the inner environment. It determines the net polarity of the membrane, right? And many other things. So how are we doing with time? Oh,
well,
we have run out of our time. Shall we stop? Or shall I continue?
So the next lecture is recorded, so maybe you can complete this slide.
Okay, yeah, so I can yeah, sure. I will complete this slide and stop. That's a good proposition. Yes. So, see what happens is, so these guys are very interesting. These guys have very important roles in lipids. Typically, lipids are considered as dumb molecules. They they just form membranes. They don't have much role and all, but that's not true. They're very interesting molecules. For example, I'll give you one example. Right, so inositol, right? So this inositol guy, what you see here, okay, so when inositol is connected to this, you have this molecule which is called as a phosphatidylinositol. Okay, now this phosphatidylinositol many times is you have different phosphate groups which are present at this further OHs. Okay, meaning that you not only have one phosphate which is connected to the inositol, you can have different phosphorelations at these OH groups. Okay. And one such molecule is called as inositol 4,5 bisphosphate. Okay, so at the position 4, so if you count this as a 1, 2, 3, 4, and 5. So you'll have a phosphate group here and a phosphate group here. Okay, now this molecule is very interesting. Okay, so what happens is, these are present in many membranes. And there are some signaling processes. So imagine there is a ligand that comes from the outside of a cell. It binds to a receptor which is called as G protein coupled receptor, okay, that are present in the membrane. And those G protein coupled receptor binding to the ligand, what happens is sometimes activates very interesting enzymes called as phospholipase. So those phospholipase, what they do is they essentially cleave off this group. Okay, so essentially here, not here, sorry, it's the carbon oxygen bond. The phosphorus oxygen bond is cleaved. So essentially what you get is, you get a molecule which has a phosphate group here, has a phosphate group here, and also has a phosphate group here. This molecule is called as IP3, inositol trisphosphate. Okay, this is a very interesting molecule because this molecule, you can imagine, it's extremely polar. And since this molecule is extremely polar, this molecule, which was initially tethered to the membrane, now it gets free. This molecule, this IP3, can travel inside cell, go to some places which are called as endoplasmic reticulum. Okay, this is a compartment in a cell. And when the inositol trisphosphate binds to the membrane or to a receptor called as inositol trisphosphate receptor that is present on the endoplasmic reticulum, it's like a gate. Okay, so it goes to that specific chamber or organelle called endoplasmic reticulum. That gate opens, and what comes out into the cytosol? Calcium molecules, Ca2+. And when calcium molecules come out into the cytosol, those calcium molecules are very, very interesting molecules because they bind to n number of proteins and they do very, very interesting things. So for example, your heart is beating because of contraction of muscles and relaxation of muscles, which is fundamentally controlled by this calcium, okay, and which is again controlled by this inositol trisphosphate. So once the calcium comes out, and then you have a huge concentration of calcium in the cell, in the cytosol, and that is directly associated with the contraction of the muscles, for example in the heart muscles. And this calcium, for example, when it's present, although through a different pathway, this calcium, when it's present in the cytosol of beta cells of islets of Langerhans in pancreatic cells, that is associated with the release of insulin, okay, which lowers your blood sugar. Okay, so yeah, so these phospholipids, essentially, depending upon the head group, they have different roles. In the next class, I'll tell you about the some phospholipids that are present on bacterial membrane and the phospholipids that are present on mammalian membrane, and how different are they, and what sort of functions do you have? Not functions, but how one can utilize it to design drugs. Yes.
Sir, I had just one doubt regarding this pathway that you told us about. So
Yes.
here the this phospholipid that has inositol 4,5 bisphosphate in its R group. Is this present in the cytosol or in the membrane?
No, it's present in the membrane because you have the acyl
Oh,
chain.
so it's present in the membrane on the inner side.
Yeah, so for example, exactly, yes, it's present in the membrane in the inner side. Okay, the other thing you might also want to know that there are I think in the next semester maybe, or in the next year, you will study that there are many enzymes that are present in the membrane which can actually flip lipids, meaning that from the outer side, outer environment, outer leaflet, they can come to the inner leaflet and all, and from the inner leaflet they can also go to the outer leaflet. Okay, yes, so in this case, yes, certainly this is present in the inner leaflet, because if it's not present in the inner leaflet, because there are certain kinases are enzymes which phosphorylate those inositol OH, which are the 4 and the 5, they also have to be phosphorylated. Okay.
Yes. Okay, sir. Okay, sir. Yes.
Yeah. Okay, I think I'm done. If you have any questions, you can ask, or you can leave.
Sir.
Yeah. So Smriti, if you're recording the class, you can probably stop recording. I think we are done.
So, sir, is this a whole pathway actually modular? As in, can this whole pathway be put into any kind of signal transduction pathway, or does this exist in only one particular thing?
No. We are talking about a very specific pathway, and you can probably clarify with Smriti. This is GPCR. This pathway is associated with G protein coupled receptors, right?
Yes.
And this is also very specific. I think G alpha or GQ, one of those receptors. Not all GPCR. There's so many GPCR.
Okay. Yes. That's why I was asking, because G protein is there in lots of different pathways. So
Yes,
this also does this also extend to all of them or something?
No. It's specific.
Okay. Yes.
Sir, and is the phosphorylation of inositol also activated by the binding of the protein to the or the molecule to the GPCR?
You mean whether this phosphorylation takes place after the ligand binds?
Yes.
This I don't know. You might want to ask someone who knows this PI3 kinase pathway. If I'm not wrong, I think there are kinases which are called as PI3 kinase, or maybe PIP2 kinases, or those. Those guys are the ones which actually yeah, definitely. It's not random. There is a signal. Only when there is a signal. I don't know I don't work in this area, so I don't know the exact pathway. But yes, I think it's triggered. Some sort of a receptor tyrosine kinase, which I think is phosphorylate. No, these are PI3 kinases. I think they're PI3 kinases. Yeah.
Okay, so is this related to the same signal transduction process as the cleavage of a PO bond is related, or is it separate from that? Or is it already like phosphorylated?
You mean, I don't know. I don't want to give you a wrong answer.
Okay. Thank you, sir.
Anything else?
But if it had been coupled to the same signal, I think it would have led to a significant time delay. Like a signal comes, it has to phosphorylate, and then it has to cut it off and then release IP3. It would significantly cause a big time delay. I don't think that might happen. I mean, I might be mistaken, but this might happen.
Okay, I mean, see, what happens is, for example, inside a cell, these processes are enormously fast and coupled.
Yeah.
Okay.
Yes.
So those time delay, we are talking about maybe fraction of seconds also. Okay.
Yes.
So as I said, I don't know, so I don't want to comment, but I just thought, you might want to appreciate the timing things. These timings are, I mean, super fast. I mean, I'll not be surprised if it's coupled. I don't know. Again, as I said, I don't know.
Thank you, sir. Thank you, sir.
Is there anything else? Okay, if there are no questions, then we will.
Sir, you once in the lecture you said that hydrogen bonds are actually not responsible for folding of proteins. So is that correct? I mean, there is hydrogen bonding between CO and NH. So doesn't it contribute to somewhat to the folding of proteins?
Well, they do contribute. I mean, it's not they do not contribute, but see the way they contribute is sometimes formation of local structures. I mean, you will appreciate this when you talk about certain structural motifs in polypeptides. There, I will probably not just remind me. Okay, at that time, I will tell you that how certain hydrogen play an important role, but those are essentially, so what happens is there is a long polypeptide chain. It's a straight line. Okay, now the polypeptide chain has to bend to fold. Okay, because it has to form a globulous shape. All right, now what happens is sometimes because of certain torsion angles, imagine that there is a bend in the polypeptide chain at the center. All right. Now it is possible, it is very likely, I mean, it actually happens also, that the bend is sort of stabilized by a hydrogen bond. Okay. Now, but okay, that local bend is stabilized by a hydrogen bond. And if you do not form a hydrogen bond, if you disrupt that hydrogen bond, the local bend will not happen. Okay. So they are important in protein folding. But how much they contribute in terms of energy, that is what I was talking. That you have to everything based on hydrogen bonds, it will be difficult to justify that so if you say that by loss of hydrogen bonds, you are stabilizing the folded state by, I don't know, 10 kilocal per mole, that will be difficult to obtain.
Okay, sir. Okay, I'm getting it.
Yeah.
So hydrogen bond energetically, its contribution is less as compared to the other factor, right?
Sorry, energetically their contribution is less, but they are important. They're certainly important.
So would it be correct to say that hydrogen bonds are not responsible for the folding of proteins, but are responsible for the stability of the folded proteins?
Yes, that would be a very nice. Yes, that would be a nice statement. That will be a correct statement. In fact, yes. Yes.
So then, as you were saying in Anfinson's dogma or Anfinson's experiment, so what then is the governing principle that allows the these proteins to spontaneously fold into their native structures?
Okay, so when we talk about that, then we will discuss. Okay.
Okay. Yes, sir. Yes, sir.
Well, definitely. This is very interesting.
Yes.
Yeah. Anything else? Okay. If there are no more questions, I'll see you guys on Wednesday. And if
Sir,
yeah,
shall we start reading Leninger of the
yes,
topics
please,
you
please.
have covered so?
Yeah, please start. I mean, for example, the forces, you will talk about water molecules. Just read. Start reading. It's always good to read. I mean, you can read, nucleic acids, proteins, polypeptides, carbohydrates, whatever you can read. Just read. It's always good to read. So if Sarah and Smriti are there, I will upload the forces part, which I took in the last and this part of this class. And I'll try to give me some time. I have to find out certain references on this chemical evolution. Don't worry, I'm not going to ask you questions. It's just for your some fun reading. Okay. All right. So thank you. Bye bye.
Thank you, sir.